chemical tests can be used to identify several important constituents of food like starch, sugars, proteins, and fat. This tutorial introduces you to the tests which can be used to detect different food constituents. If you like what you see on our channel, please like, share, subscribe, and turn on notifications. The presence of starch, a complex carbohydrate in a food sample, can be determined using iodine solution. Iodine solution is yellow or light brown, but it turns blue-black in the presence of starch. As an example of this process, let's place a small piece of potato onto a white tile. Add 2 to 3 drops of dilute iodine solution on it. Now observe the changes. The piece of potato turns blue-black in color. This is a positive test which proves the presence of starch. If starch was absent, the original yellow or light brown color of iodine solution remains unchanged. This test can also be performed using a liquid food sample. Now let us test food for the presence of reducing sugars. Do you know what are reducing sugars? Simple carbohydrates usually containing 1 or 2 sugar units which act as reducing agents are called reducing sugars. Example: glucose, fructose, lactose, etc. Benedict's solution is used to check for the presence of reducing sugar in a food sample. Cut or grind some food, place it in a test tube, add water to it and shake it. To this liquid food sample, add some bright blue colored Benedict's solution. Heat the test tube to about 80 degrees Celsius in a water bath. You will observe that the mixture changes gradually from blue through green, yellow and orange, and finally brick red. Benedict's test can be used to know how much simple sugar is present. If the color changes to green, the extract only contains a little of the reducing sugars. If it goes a deep orange color, then it contains a lot of reducing sugars. If there is no reducing sugar, then the Benedict's solution remains blue in color. Stay tuned as we continue to test foods for protein, fat, and vitamin C in our next tutorial.